everyone. This is the lecture on membrane structure and function. This comes from Campbell Biology Chapter 7, and if you happen to be using the OpenStax textbook, this corresponds to Chapter 5. So how do things get in or out of the cell? It depends. It depends on the size of the molecule and then other chemical characteristics of the molecule. So small molecules, some can move across the cell membrane and go into or out of the cell using what's called passive transport. And so there's no input of energy. This can happen on their own. The molecules can come in and out, or they may require transport proteins for help to get in or out of the molecules or the plasma membrane. Some other molecules use active transport, and this kind of transport requires energy and is transport protein. So that's for small molecules, and then for large molecules, it has to happen a different way. So in this case, molecules will move in and out of the cell using what's called bulk transport, either exocytosis or endocytosis. So that's kind of the overview that we're going to talk about throughout the lecture. So cell membranes are formed from phospholipids. So again, if you remember from the carbohydrates and macromolecules, lipids. So lipids are a class of molecules, and phospholipids are one kind of a lipid. And so they have two parts to them. So phospholipids actually are amphipathic, meaning they have some parts of the molecule that like water and some that don't. So the hydrophobic tails of the phospholipids will end up facing the inside of the membrane, and then the hydrophilic heads will face the outside of the membrane. And remember that that's based on if they're polar or non-polar. So if you were to put phospholipids into like a cup of water, they would spontaneously form this lipid bilayer. So bi meaning two, so there's one layer of lipids and then the other layer. And then they orient so that they have the tails facing each other, and then the hydrophilic heads interacting with the watery environment outside of the cell and then inside of the cell. And so that means that most membrane proteins, or like proteins that are in the membrane, also have to be amphipathic. They also have to have parts that are hydrophilic and parts that are hydrophobic, so that they can hang out in those specific areas. So the hydrophilic parts of the protein are going to be oriented toward the cytosol and the extracellular fluid inside and outside of the membrane. So yeah, the parts that like the watery environment are going to face the watery environment outside the cell and inside the cell. And then the hydrophobic regions of these proteins are going to be inside the bilayer. And remember, there's very complex structures of proteins. We just show them as blobs because it's kind of easier. But remember, proteins are long chains of amino acids, and there's regions that are hydrophobic and hydrophilic, depending on what amino acids are there. So this is a picture showing the cell membrane. Again, even though you might see in like a depiction of a cell, it's just like one, it looks like one layer. It's actually the two layers of the phospholipids. And there's all kinds of stuff embedded in that's not always shown in figures of the cell. So this is what's called the fluid mosaic model. And this shows that there is a mosaic of things that are in the membrane. So there's phospholipids, and then there's a lot of different kinds of proteins that float around in the membrane. And so if you were to look from the outside, it kind of would look like a mosaic. Let me show a picture really quick. So this is a mosaic in case, it didn't come to mind immediately. It's by like a style of art where you put pieces of different kinds together. So a membrane is kind of like that. It's a collage of different proteins and they tend to be clustered in groups, and they are embedded in the lipid bilayer. So this is what's called the fluid mosaic model. So you can kind of see how it looks like that. And the proteins are not randomly distributed. They don't actually make a picture, literally, but they are grouped together. The proteins are grouped together if they carry out common functions. So maybe like this is for one function, and these all interact for a different function. Yeah, so there's a lot going on that we can't really show in every single slide or every single figure of a cell, but there's a lot going on. So the membranes themselves are held together mainly by weak hydrophobic interactions. Most of the lipids and some proteins can move sideways within the membrane. So they're not just stuck in these positions. It's not like rigid. It's kind of fluid. Things can move around. So most of the lipids and some proteins can move sideways within the membrane. And this is important. Membranes have to be fluid to work properly. They can't be like rigid and stuck in place, or your cells can't really be, I don't want to say adaptable, but kind of like, or else they can't function the way they need to and alter things. So, temperature can make a difference in the fluidity of membranes. As temperatures cool, membranes tend to switch from a fluid state to a more solid state. And the temperature at which a membrane solidifies depends on the types of lipids. So is it, are they saturated fatty acids or unsaturated fatty acids? That makes a difference structurally and then also like functionally. So membranes that are rich in unsaturated fatty acids have those like bumps in the tails. And so the molecules can't pack as tightly together. And so these membranes with these kinds of tails, unsaturated, are more fluid than those that are rich in saturated fatty acids. So here on the right, you see phospholipids with saturated fatty acids. And so they're really tightly packed together, and they're more likely to be like solid at certain temperatures than ones with unsaturated tails. Another important component is cholesterol. So I think there's a picture on the next slide. Yes. So cholesterol is a membrane component in animal cells, and it can have different effects on membrane fluidity at different temperatures. So sometimes it helps keep the membrane more fluid, and sometimes it helps keep it more solid. It's kind of adaptable that way. So at relatively high temperatures, cholesterol restrains movement of phospholipids, so it keeps it a little bit more like solid. It prevents getting too liquidy. And then at low temperatures, it maintains fluidity by preventing tight packing. So it serves dual purposes depending on temperatures. And so cholesterol is in animal cells, cell membranes, and then plants use different but related steroids, steroid lipids, to help buffer that membrane fluidity. So here's a picture of the cholesterol molecules and then how they're embedded in the phospholipid bilayer. And again, membranes have to be fluid to work properly. Having the fluidity affects both the permeability, or like how well things can go through, and the movement of transport proteins, whether or not the proteins can be moved around and like added or removed. So membranes that are too fluid can't support protein function. So you have to find, like a nice kind of buffer zone. And it's really interesting that organisms that live in extreme temperatures can have adaptive differences in their membrane lipid composition. So for example, the lipid makeup of the cell membrane appears to be adapted to the environmental conditions in many species. So fish that live in extreme cold usually have cell membranes with a high proportion of unsaturated hydrocarbon tails. So remember, those are the ones with like the little bends. And so again, having those bends keeps it more fluid at these cold temperatures and not getting too rigid and solid. And so that's organisms that live in an extreme environment and they never really leave that environment. They had to adapt to have something in their membrane that helped in that environment. Some organisms live in variable temperatures. So, you know, areas with different seasons where the temperature goes to like, fluctuates a lot. So organisms living in variable temperature conditions can change lipid composition in response to those changing temperatures. So for example, in winter wheat, the percentage of unsaturated phospholipids increases in autumn to prevent membrane solidification during winter. Biology is so cool. So again, back to the idea of membrane proteins. So a membrane is a collage of different proteins, often clustered in groups, embedded in that fluid matrix. And so, yeah, they say it kind of looks like a tile mosaic. And so the proteins that are in a membrane help determine most of the membrane's functions. So again, like different cells that have different special functions are going to have some slightly different variations of cell structures. So depending on what a cell is supposed to do will depend on what proteins are in the membrane. So the protein makeup of membranes can vary among cells within an organism and even among intracellular membranes within the same cell. So one cell in your body isn't always going to have the same proteins in the membrane as another cell. And then even in the same cell, there might be different proteins on one side of the cell versus another side of the cell. Which is especially, makes sense, especially if you think of like the neurons that are around my background. These are specialized cells that one end are probably going to have receptors. And then at the other end might have proteins that help communicate to the next cell. And so there are two major types of membrane proteins based on location. So these terms are more about like just describing the location, not necessarily what they do. So peripheral proteins are on the periphery of the membrane. They're on either side, like inside or outside. They're on the surface. They're on the periphery. So peripheral proteins are bound to the surface of the membrane. And integral proteins are integrated in the membrane. They penetrate that hydrophobic core. And so, this is an integral protein, but then so is this one because it, penetrates the core. So those are two terms just kind of based on location. And then one more, transmembrane proteins. These are integral proteins that span the whole membrane. So it reaches from one side to the other. And these kinds of proteins are able to do this because they have parts that are hydrophilic and then parts that are hydrophobic. And so those hydrophobic regions on the inside consist of non-polar amino acids, and they're often coiled into the alpha helices. So again, even though we show proteins a lot of times as blobs, there's a lot of structure going on there. And that is what gives kind of the shape to the blobs. And so some membrane proteins are held in place by attachment to the cytoskeleton inside the cell. Some other proteins attach to materials outside of the cell, like to the extracellular matrix. So there are some proteins called integrins that attach to fibers of the extracellular matrix. Let me go back to this one. So for this protein, you can kind of see there is a protein and then there is another protein that's anchoring it to the extracellular matrix. Or you can't really see exactly a point of connection, but yeah, some of these proteins are connected to the cytoskeleton. Okay, and so there's all kinds of membranes in the proteins in the membranes, and they can do several functions. They can help transport things in and out of the cell. They can function as enzymes and perform or help speed up some kind of chemical reaction. Signal transduction, meaning like when cells communicate to each other, it like passes on the signal inside of the cell, so that that action can be carried on from the message that it got from the previous cell. Membrane proteins can help in cell-cell recognition. So depending on what protein's there, it helps another cell know, oh, okay, you're a fellow skin cell. It can help with intercellular joining to help keep tissues together. And then again, it can also attach to the cytoskeleton and extracellular matrix. So they can do all kinds of different things. Proteins, again, are super varied. So here's some pictures showing each of these different functions. So here are different kinds of transport proteins that we'll talk about a little bit later. So they help certain things get in or out of the cell. Some membrane proteins can function as enzymes, and so they are speeding up this chemical reaction, and then maybe this one's doing this chemical reaction. A signal transduction pathway would have, a signaling molecule bind to a receptor, and then this receptor would send some signal and pass on that message. Cell-cell recognition based on what's attached to proteins that can help other cells know, okay, we're working together. Intercellular joining, and then again, this picture was here. Attachment to the cytoskeleton on the inside, and extracellular matrix. So it just depends on the protein, really. And so, sometimes you might be taking this class for some other bigger picture career, but it is important. All this stuff that we learn in this class in cell and molecular bio is relevant to all of us and all branches of the field. So for example, cell surface proteins are really important in medicine. So in this example, we are considering HIV, the human immunodeficiency virus. And so this is, a cartoon of the virus. And then notice the size of the virus compared to the whole cell. The cell's a lot bigger. Again, viruses are not real cells. They're not alive. They don't have like a cell membrane. They're super tiny. They don't have organelles, etc. And so on the surface of virions, there are little proteins that stick out. And they happen to resemble some of our, the molecules in our cells. And so they will kind of trick our receptors into taking them in. So HIV enters immune cells by binding to cell surface proteins. So there's one receptor called CD4 and a co-receptor called CCR5. And so together they happen to recognize this. This virus protein tricks it into taking in the virus, and then it enters our cells. But if individuals are lacking that co-receptor, then it's not really functioning to end up getting tricked and bring those things in, bring the viruses in. So yeah, individuals lacking that protein, that receptor, are immune to HIV infection. And so drugs are in development to kind of see if we can mask CCR5 and block HIV's entrance in non-immune individuals. So again, it pays to learn about all these concepts. It does all apply, even if you don't think it does. And so cells, as I mentioned, can recognize each other by binding to molecules on the surface of the membrane. And many of the surface molecules are bonded to short branched chains of carbohydrates. So back to this picture. You can see some proteins have stuff sticking out. So a glycoprotein has carbohydrates sticking out. A glycolipid is a phospholipid with a carbohydrate sticking out. So there can be other things attached to parts of the membrane. Yeah, so that's what helps cells identify each other, is based on what's sticking out. So glycolipids, glyco usually refers to carbohydrates. So glycolipids are lipids that have carbohydrates stuck on. Glycoproteins are proteins that have carbohydrates stuck on. And so having this wide diversity of the surface carbohydrates can help them function as markers for cell identification. Because even if there's different glycoproteins, they might not all be the same carbohydrate. So having different ones there helps, serve as markers for cell identification. And so the membrane, the cell membrane, but also the membranes on the inside of the cell, they have distinct inside and outside faces. So that's how the cell kind of knows what's what and what's where it's supposed to be where. So the makeup and distribution of proteins, lipids, and carbohydrates is going to be different around the membrane. And then even in these little membranes inside the cell.